Hi, my name is Janae Heiss, and I am a student at Frostburg State University. I'm making this video on the topic of photosynthesis for my master's degree in applied ecology and conservation biology. My thesis for this degree is on cannabis cultivar chemotype and genotype. In basics, that just means how different types of cannabis look genetically and how they look in their chemical composition. This video ties into my master's project because cannabis is a plant, and plants do photosynthesis. In addition to that, my research uses Rubisco as a test to make sure my samples have amplifiable DNA. This video, per Dr. Podol's request, is 60 minutes long and does not use slides or notes. Hi everyone! In this video, I will be covering an overview of photosynthesis, the different parts of plants, some basic parts of leaves, and chloroplast structure, as well as some important historical figures in the history of photosynthesis. Light and the scientific properties of light, pigments and accessory pigments, and then the light reactions with the antenna receptor complex, the electron transport chain, and Atp Synthase, and the Calvin cycle with its three main phases: fixation, reduction, and regeneration. And lastly, I will be covering the main ways that photosynthesis and cellular respiration differ. Today, we will be talking about photosynthesis. So, photosynthesis, what this video is all on. Photosynthesis is from the Greek words photo, which means light, and synthesis, which means combining simple things together to make something more complicated. So, we're using light to turn something simple into something complex. It's taking carbon dioxide and using them to make complex things like sugars. Photosynthesis converts solar energy, or light, into chemical energy, or molecules. Organisms can be classified into two main categories according to the way that they make their energy. And this may have, if you saw the video on cellular respiration, we've gone over this there too. So there are autotrophs and heterotrophs. Autotrophs, from the Greek self-nutrition, and they make their own nutrition, like plants, which is what this video is on. And then there's heterotrophs, and they have other nutrition. They eat other things, like plants or animals or anything else, to get their own nutrition. So, the autotrophs make their own energy, and photosynthesis is how those autotrophs do it. Alright. The formula for photosynthesis is this: 6 CO2 plus 6 H2O, so 6 carbon dioxide and 6 water, come together to make 1 sugar molecule, in this case glucose, and 6 oxygen molecules. This is the basic formula for photosynthesis. It gets much more complex than this, but this is it in basics. And this is also the reverse of the equation for cellular respiration. So instead of using sugar and breaking it down to make carbon dioxide, we're taking carbon dioxide and building it up to make sugar. Photosynthesis is really important. Despite only 1% of the energy from the sun being captured by photosynthesis, the process of photosynthesis is still used to provide the energy needed for all the living things on Earth. So it's a really important process. Photosynthesis has two main parts: the light reactions and the Calvin cycle. Photosynthesis mostly occurs in the leaves of plants, in the mesophyll region. Meso is middle, so it occurs in that middle part. Plant cells have chloroplasts, and those chloroplasts have an important green pigment called chlorophyll inside them. The chlorophyll pigment absorbs light, and then the light is used to make ATP. Each mesophyll cell has about 30 to 40 chloroplasts on average, and chlorophyll pigment is found in the membrane of the thylakoids. On the underside of the leaf, there are many structures called stomata. The stomata open and close to allow carbon dioxide in and oxygen out. Each stoma, the singular of stomata, has a pair of guard cells there that surround it and control whether the stoma is open or closed. Chloroplasts have many internal structures. From the outside in of the chloroplast, there is the outer membrane, and then the intermembrane fluid between the membranes, also called the intermembrane space, the inner membrane, and then a dense fluid called the stroma. The stroma is where the Calvin cycle takes place, the second part of photosynthesis. Also in the chloroplast are the thylakoids. Thylakoids are like stacks of membranous sacs. They look like a green interconnected highway of pancake mounds. So we've got these stacks of pancakes, these stacks of thylakoids, and we have the stroma lamella connecting them. And those are the highways connecting them. The thylakoid membranes are the place for the light reactions, the first part of photosynthesis. And then the area inside the thylakoids, the thylakoid lumen, is used to hold the proton gradient in the light reactions. So both the thylakoid membrane and the thylakoid lumen are really important for the light reactions. In the light reactions, energy from the sun is made into ATP. In the chloroplasts, there's the thylakoid membrane, which is the membrane of those pancake-like discs, and that thylakoid membrane has photosystems in it. Those photosystem molecules are hit by the light, and it energizes molecules in those photosystems. The light does. And then when water comes into the area, the photosystem takes electrons from the water and reduces that water into oxygen and hydrogen ions. So the oxygen gas leaves the area at the stomata, and the products eventually of the light reactions are ATP and NADPH. The way ATP is generated from ADP in the photosynthesis is called photophosphorylation. This is a specific type of phosphorylation because we're using it in the light reaction. So we have photo, light, in front of it. In the Calvin cycle, carbohydrates are made from carbon dioxide. And a lot of energy is spent in order to make those carbohydrates. The energy that they use is in the form of ATP and NADPH. Those are the things we got from the light reactions. So we take the energy we made in the light reactions and we use it to do the Calvin cycle. Carbon dioxide goes into the Calvin cycle and sugar comes out. NADP plus stands for nicotinamide adenine dinucleotide phosphate. Like the NAD plus in cellular respiration, NADP plus is also cycled. The phosphate group separates those two from being the same molecule. So NADP plus cycles between NADP plus in the Calvin cycle and NADPH in the light reactions. So what happens is NADP plus goes into the light reactions, gets turned into NADPH, NADPH goes to the Calvin cycle, and then gets turn back into NADP+. So it just cycles between the two. Before we go any further, there's some history background to cover. So photosynthesis began with a man named Baptista Van Helmont. Most people have already either heard of this man or the work that he did. Van Helmont lived from 1580 to 1644. That's around 400 years ago. And he discovered the first property of photosynthesis. To do that, he took a willow tree and then he weighed it out. And then he took some soil and he weighed it out. He planted that willow tree in the soil and let it grow for 5 years. And then he took the tree from the pot and discovered that while the soil weighed about the same that it did in the first place, the tree had gained weight. So he discovered that the plants gain weight not from the soil, which was previously thought, but from somewhere in the atmosphere. So something in the atmosphere they took in and it caused it to gain mass. Joseph Priestley is another important figure in the history of photosynthesis. He liked candles and glass ball jars. To start, Priestley took a candle and he put a glass ball jar over it. Inevitably, the candle would go out because it used up all the oxygen in the jar, and so it couldn't burn any more. So then, Priestley put a mint plant under that ball jar with the candle, and this time the candle stayed lit. And it stayed lit for a long time. So the plant supplied something to the ball jar that allowed the candle to stay lit. We learned that what it supplied was oxygen. The plant produced enough oxygen that when Priestley decided to put a mouse under there, in that same ball jar with the plant and the candle, that mouse stayed alive, the candle stayed burning, and the plant stayed alive. So through this, we learned that plants supply oxygen to their environment, and that oxygen they supply helps animals live. Plants supplying oxygen to their environment is an important part of how life survives on Earth. In addition to Jean-Baptiste van Helmont's 1641 experiment and Joseph Priestley's 1771 discovery, there are also other notable scientists in the history of photosynthesis. After Joseph Priestley's experiment with the bell jar, in 1779, a Dutch physician, Jan Ingenhousz, demonstrated that plants are only able to provide gas to the air in sunlight, not darkness. So he took a bell jar and he put it in the dark and determined that the plants in the bell jar were not able to continue photosynthesis, and therefore the organism in the bell jar was killed. So Ingenhousz demonstrated that the green parts of the plant are needed for the gas to be released, and sunlight by itself was ineffective at causing whatever gas it was to be released. The French chemist Antoine Lavoisier demonstrated that candles and animals both consume a gas in the air. He named this gas oxygen, and that's how oxygen has its name. He determined that the gas that was produced in the earlier bell jar experiments was actually oxygen. He also named hydrogen gas. In addition, Lavoisier's work showed that water was not an element, as was previously stated. He completely disproved the earlier elemental theory, which had earth, wind, fire, and water as the main elements. He determined that water was not an element, it was a combination of elements. And those two elements were hydrogen and oxygen. He proved this in 1784. Lavoisier revolutionized the science of chemistry, and he transformed it into its current quantitative form. He also is responsible for a lot of the nomenclature in chemistry, such as sulfate and sulfite. Though Lavoisier is considered to be the founder of modern chemistry, during his lifetime he was condemned. So he was condemned to death and beheaded, as one of the many figures were during the French Revolution in 1794. In 1804, Nicolas Théodore de Saussure showed that the increase in the mass of a plant as it grows was not just due to the uptake of carbon dioxide, but was also the result of water. So plants need not only carbon dioxide to grow, they also need water. He demonstrated this by showing that plants grown with their roots in water and their shoots in a carbon dioxide-rich atmosphere increased in their weight much more rapidly than expected from carbon dioxide alone. The increased weight had to come from water. Saussure also showed that plants require mineral nutrients, which they take up from the soil. So he was the one that determined what plants take up from the soil. Saussure's work enabled the completion of the basic overall chemical equation for photosynthesis. His work showed carbon dioxide and water result in sugars and oxygen being produced. The Dutch-American microbiologist Cornelius van Niel determined what the oxygen from photosynthesis was actually derived from. Van Niel studied photosynthesis in anaerobic bacteria instead of in plants. Like plants, photosynthetic bacteria make carbohydrates during photosynthesis, but bacteria do not produce oxygen during photosynthesis, the bacteria he was using. They also used a different set of photosynthetic pigments than plants do. In his 1931 experiment, Van Niel found that all species of the photosynthetic bacteria he studied required an oxidizable substrate. For example, his purple sulfur bacteria used hydrogen sulfide as the oxidizable substrate. Based on his studies with photosynthetic bacteria, Van Niel proposed that the plant's oxygen produced during photosynthesis was from water, not carbon dioxide, because water was the oxidizable substrate. In the following years, this hypothesis was supported by the work of Robert Hill. Robert Hill, also known as Robin Hill, found that oxygen produced during photosynthesis was actually made during the light reactions. In 1937, he developed dyes that enabled him to determine the electron transport chain configuration, and this in turn helped him to determine that oxygen was produced from the electron transport chain. So, the oxygen was produced from a protein in the electron transport chain reacting with water. In the 1950s, Hill developed the Z-scheme of electron transport through the light reactions. The study of photosynthesis is still an active area of research in biology. In the 1980s, Hartmut Michel and Johann Dieselhofer made an important contribution to our understanding of photosynthesis. They made crystals of the photosynthetic reaction center from a bacteria that was anaerobic, and then they used X-ray crystallography to determine the structure of that reaction center. They shared the 1988 Nobel Prize in Chemistry with Robert Huber for this groundbreaking research. So, the history of photosynthesis has come a long way from the 1700s, where people thought that plants ate the soil. And now we know all about the structure of photosynthesis and the different proteins that are responsible for it. Light is an important concept in understanding photosynthesis. Out of all the wavelengths of light, we can only see a small fraction of them, known as visible light. Visible light is separated into different wavelengths too. Red is the longest wavelength of light, at 620 to 750 nanometers. Then we have orange light, yellow light, green light, blue light, and indigo light. Violet is the shortest visible wavelength, at 380 to 450 nanometers. Pigments are specific molecules that absorb light. Because the plant wants to absorb as much light as possible, it's advantageous for the plant to have different types of pigments. And whatever color that is not absorbed by the plant is reflected. In the case of chlorophyll, the main pigment in plants, chlorophyll absorbs every wavelength except green. So green light is what's reflected. And this is why plants appear green. But chlorophyll isn't the only pigment in plants. There are also accessory pigments. Accessory pigments are secondary pigments that absorb the wavelengths that chlorophyll does not. There are carotenoids, which appear orange, and xanthophylls, which appear yellow. There are also other pigments in plants, but those two are the most common. Carotenoid molecules and other accessory molecules give leaves their color in the fall. They also act as important antioxidants to protect the plant. They stop reactive oxygen species from forming. There is more than one type of chlorophyll pigment. Chlorophyll A has the highest absorption at 430 and 660 nanometers. Those are purple and red wavelengths respectively. Chlorophyll A has the best absorbance in the colors red, blue, and purple. And chlorophyll A is the main pigment for photosynthesis. Chlorophyll B absorbs best in the red, orange, blue-green, and blue wavelengths. It absorbs best at 450 and 640 nanometers, and those are blue and orange wavelengths. The different types of pigment molecules allow the plant to absorb more light energy, and the plants take that light energy and they convert it to chemical energy. So the more light energy they absorb, the more chemical energy they can produce. Chlorophyll molecules have a large head group with many rings and a long tail. In the center of the head group are several nitrogen atoms surrounding a central magnesium atom. Plants are actually fluorescent because of their chlorophyll molecules. So back to photosynthesis. First are the light reactions. The light reactions are where sunlight is used to make ATP. In the thylakoid membrane, there are photosystems which span across the membrane. These photosystems are full of hundreds of pigments arranged in an antenna complex. The light hits the photosystem and energy bounces around the pigments until it hits that reaction center chlorophyll. So we have the antenna complex, we've got the pigments arranged around the antenna complex, and then the reaction center chlorophyll in the middle. The light hits one of those pigments, it bounces around on the reaction in the antenna complex, and then it eventually reaches that center reaction center chlorophyll. That reaction center chlorophyll is chlorophyll A. Chlorophyll A loses electrons, and then those electrons are passed on to other molecules through an electron transport chain. In this step, light photons are converted into energy. And this is really important. It's really cool too, because light energy is transformed into chemical energy in this step. The light photons hit the pigments, the photons bounce around into the center chlorophyll A and energize it, and then that energy is transferred to chlorophyll A. And in response, that chlorophyll loses electrons. To replenish the electrons that were lost, water is reduced, and those electrons are donated back to the chlorophyll. And because water is reduced and it loses electrons, it makes oxygen gas and hydrogen ions as the byproducts. The hydrogen ions stay in the thylakoid lumen, but the oxygen gas is released out of the stomata and goes out of the plant. The order that the electrons flow through the membrane is through Photosystem 2 and then Photosystem 1. Photosystem 2 is the first photosystem in the sequence, and then Photosystem 1 is the second photosystem in the sequence. The two photosystems were named in the order that they were discovered rather than the order that the electrons actually passed through them. Photosystem 2 has a P680 primary donor, so that's the type of wavelength that it has the maximum absorbance at. The whole light reaction flow is that light hits photosystem 2, two electrons from photosystem 2 flow into the electron transport chain, and then they flow through the electron transport chain through a molecule called cytochrome Bf. Then, after another molecule in the electron transport chain, the electron goes into photosystem 1. Photosystem 1 has a P700 primary donor, so it absorbs best at 700 nanometers wavelength. Light hits photosystem 1, and the excited electrons flow into more molecules in that chain. The electrons flow into NADP+. NADP+ takes those two electrons and the hydrogen ions and is converted into NADPH. NADPH is going to be used in the Calvin cycle. Despite being similarly structured to Photosystem 2, Photosystem 1 is dependent on other earlier parts of the chain to get electrons. Photosystem 1 receives electrons from plastocyanin, one of the molecules in the electron transport chain. And then, after the light excites those electrons in photosystem 1, the electrons are transported to ferredoxin, which is another molecule in the electron transport chain. Because the electron transport chain energizes and then the electron is unenergized and then it re-energizes that electron, it's typically called the Z scheme. Because the electrons flow into photosystem 2, through the electron transport chain losing energy, through photosystem 1, and then they lose energy and go to NADPH. In addition to making NADPH, the electron transport chain also pumps protons across the membrane. Protons are pumped into the thylakoid lumen, which is the innermost layer of the chloroplast. Atp synthase pumps those protons across the membrane using photophosphorylation. ATP synthase takes the ADP and makes ATP. And this whole process of the light reactions requires sunlight, because without light, those photosystems would not be able to get excited and they would not be able to transport electrons from one molecule to the other. The electron transport chain in photosynthesis is very important. So let's go through step by step each of the molecules in the electron transport chain and what they really do. So we start with photosystem 2. Photosystem 2 has an excited P680 reaction center chlorophyll. And then it passes those electrons to phaophyte. Photosystem 2 gets more electrons after it passes them by splitting water into oxygen gas and hydrogen ions. So the electrons go into photosystem 2 and they get energized. Then from photosystem 2, they get passed to pheophyton, losing a little bit of energy. Pheophyton is inside the photosystem 2. After pheophyton gets those electrons, it passes them to plastoquinone. Plastoquinone passes those electrons to cytochrome Bf, and cytochrome Bf transfers those electrons to plastocyanin. Then plastocyanin donates the electrons to the P700 reaction center in photosystem 1. Photosystem 1 re-energizes that electron, making it have more energy. And then, from there, it passes that electron to ferredoxin. Ferredoxin passes that electron pair to ferredoxin nucleotide reductase. And ferredoxin nucleotide reductase takes those electrons and finally transfers them onto NADP+. NADP+ is then reduced into NADPH, and it can be used to make to fuel further reactions down the line. And this whole process of the electron transport chain transfers those electrons so they can be used to power those reactions that are going to be going on during the Calvin cycle. To summarize everything that's happened in the light reaction so far, we've got 4 main steps. Step 1. A photon of light is captured by a pigment molecule and bounced through the photosystem. Step 2. Energy is transferred to the reaction center and the electron is transported to an acceptor molecule. Step 3. The electron transport chain moves the electrons through the chain and reduces NADP+ to NADPH. And through that electron transport chain, photons are pumped through the thylakoid membrane. And then step 4, chemiosmosis produces ATP through ATP synthase. After the light reactions, the second phase of photosynthesis is carbon fixation. This is through the Calvin cycle, which is also called the light-independent reactions. The Calvin cycle is sometimes also called the dark reactions, but the dark reactions is a misnomer. It does not need to be dark for the organisms to go through the Calvin cycle. It can be either light or dark. It doesn't matter. This cycle takes place in the stroma, the middle layer of liquid in the chloroplast. There are 3 main phases to the Calvin cycle, and the point of the Calvin cycle is to take carbon dioxide and turn it into a 3-carbon sugar. So, 3 main steps to the Calvin cycle. The first phase is carbon fixation. In this step, ribulose bisphosphate, also called RUBP, has CO2 added. And this is typically done in a replicate of 3. So 3 RUBP molecules have 3 CO2s added to them. And this is known as carbon fixation. It forms a 6-carbon molecule that quickly gets broken down into 6 3-carbon molecules. So we end up with 6 3-carbon molecules of a molecule called 3-PGA. The enzyme that converts this whole thing, converts RUBP into 3-PGA, is called Rubisco. Rubisco stands for ribulose bisphosphate carboxylase oxidase. The carboxylase is because it takes and carboxylates RUBP, adding the carbon dioxide to it, And then the oxidase is because it also acts to oxidize molecules. The next step for this whole cycle of the Calvin cycle is called reduction. In this, PGA, 3-PGA, is reduced to G3P. And this is through a series of reactions. It uses the ATP and the NADPH that were formed during the light reactions. 6 ATP and 6 NADPH are required to make the 6 G3P. Only one G3P molecule actually exits this cycle. The remaining 5 molecules of G3P stay and are used to go through the last step. The last step of the carbon cycle is called regeneration. And this is because we regenerate those RUBPs that were used in the beginning here. So we take those 5 G3P molecules and we take it and regenerate RUBP using 3 ATP molecules. Then the whole cycle can repeat. 3 molecules of carbon are required to make 1 molecule of G3P. And G3P is then used to make other molecules. 2 G3Ps can come together to make 1 glucose molecule. G3P is the direct product of the cycle. But when we're talking about the main equation of photosynthesis, we typically take and put 2 G3Ps together to get that glucose to give us that nice, simple equation. In more detail for the Calvin cycle, 5 RUBP molecules, or 5 ribulose bisphosphate molecules, each get carbons attached to them. This makes an unstable 6-carbon molecule called 2-carboxy-3-ketoarabinitol 1,5-bisphosphate. And that fun-named molecule splits in half to make 2 3-carbon molecules of 3-phosphoglycerate. So 2 3-carbon molecules of 3-phosphoglycerate are made for every RUBP. The enzyme that adds the carbon to RUBP is called ribulose bisphosphate carboxylase oxidase, or Rubisco. In reduction, the next step, some ATP and NADPH are used. 3-phosphoglycerate gets a phosphate from the ATP and becomes 1,3-bisphosphoglycerate, and then 1,3-bisphosphoglycerate is reduced to glyceraldehyde 3-phosphate. And then lastly, in regeneration, there is a series of molecules. And because there's so many different ones, we're just going to get to the main number of carbons that each molecule contains. So it starts with the G3P molecules coming together to form a 6-carbon molecule, and then that 6-carbon molecule and a 3-carbon molecule join together to form a 9-carbon molecule. But that 9-carbon molecule is unstable, so it splits up into a 4-carbon molecule and a 5-carbon RUBP molecule. That RUBP molecule is what we want, because we want to regenerate the RUBP. So we need to make another of those. So we take more G3P molecules and we regenerate RUBP using them. So we take that 4-carbon molecule that's left over, we add another 3-carbon G3P molecule, and then another to make a 10-carbon molecule, and then that 10-carbon molecule splits up into 2 more 5-carbon molecules. So we end up with 3 5-carbon molecules to end all of that. And that's how we regenerate the 3 RUBP that we need for the Calvin cycle. Photosynthesis is responsible for generating new carbohydrates from CO2, and it's responsible for the oxygen in the atmosphere. Both carbon dioxide, fixation into carbohydrates, and oxygen are vitally important for life on Earth. The concepts of cellular respiration and photosynthesis are similar, so it's important to make sure the differences between each of them are made clear. So let's take a look at those. First, there's a difference where each takes place. Cellular respiration takes place in the cytoplasm for glycolysis, and then it moves into the mitochondria for the rest of the steps of cellular respiration. It uses the inner mitochondrial membrane for the electron transport chain, and it builds up protons in the intermembrane space, which is the middle space of the mitochondria. Photosynthesis, on the other hand, only uses the chloroplast. There isn't any other area of photosynthesis that takes place in the rest of the cell, so it's only in the chloroplast. It uses the thylakoid membrane, which is the innermost membrane structure for the electron transport chain, and then it builds up those protons and the thylakoid lumen, which is the innermost space of the chloroplast. The second main difference for photosynthesis and cellular respiration is what organisms are able to do each. So photosynthesis, only plants can do photosynthesis. Plants, algae, and some bacteria. It's pretty specific. These organisms make their own energy and they store it through photosynthesis. Now, cellular respiration, on the other hand, is a global process. Every living organism at least does glycolysis to make some form of energy. Many go through some sort of aerobic or anaerobic respiration. Plants, animals, fungi, protists, bacteria, everything needs to use some sort of cellular respiration to make the energy that they need to live. And the last main difference between photosynthesis and cellular respiration is what are they used for? Cellular respiration is used to make energy for cells to use in other processes. It breaks down glucose and other sugars to make ATP. So cellular respiration is used for energy production. It's used when organisms break down chemical energy and use it to fuel other reactions in their bodies. Photosynthesis is used to make that energy. So it stores the energy up in as chemical energy. So it takes the energy from the sun, stores it up as chemical energy, as glucose and other molecules, and then it uses it later in cellular respiration. So photosynthesis uses light reactions to make ATP and the Calvin cycle to store that energy as sugar molecules like glucose. Both photosynthesis and cellular respiration are important processes. Those two processes form a big cycle across all organisms, and it helps transform energy into the forms that we need to live. Let's look at some other factors in photosynthesis. There are three main things needed in photosynthesis: water, carbon dioxide, and light. The limiting factor in photosynthesis is typically water, because plants need water the most. They have to bring it up from the roots. Light and carbon dioxide are usually in plentiful supply because of the atmosphere and the sun. The secondary limiting factor is either light or carbon dioxide, depending on the temperature and conditions of the world around them. Other limiting factors in photosynthesis are in the Calvin cycle. The main factors in the Calvin cycle are how fast can Rubisco fix RUBP and CO2 together to make that 6-carbon intermediate that's unstable and turns into the 3-carbon intermediate, and then how fast can RUBP be regenerated from G3P. The plant has different needs, and if it needs to make more structural molecules, it can siphon G3P out of the cycle to make starch or other molecules. If the plant has energy needs, it can use photosynthesis to make more energy, use those light reactions, and then use less of the Calvin cycle and just take the fats and sugars from that to get the NADPH to NADP+, so it continue the light reactions. The cycle is dynamic, so whatever the plant needs, it can modify the cycle to do. With more RUBP, the plant is able to capture more CO2. With more Rubisco enzymes, the plant is also able to capture more CO2. So if the plant has good functional Rubisco enzymes, it can capture more CO2 than a less functional version. It's a balancing act between the amount of G3P that's being taken out of the Calvin cycle and the amount of RUBP being put into the Calvin cycle. There are different factors that are determining whether the plant is able to collect enough CO2, carbon dioxide, or get enough light. Because stomata need to close to conserve water in warm temperatures, carbon dioxide could be cut off if the temperature is too hot. If the weather is too cloudy or stormy, the plant may not be able to get the light it needs to do photosynthesis. In addition to weather problems, the position of the leaf on the plant could determine whether the leaf is able to get enough light to do photosynthesis. Leaves at the bottom middle of the plant may not be able to get enough light to even do photosynthesis at some times. When determining the amount of light that's able to reach the plant, one needs to consider irradiance, specifically photon irradiance. Photon irradiance is the number of photons in a specific area per second, and irradiance is the amount of energy in a specific area per second. So researchers measure the photon irradiance that is reaching a specific leaf to determine the amount of photosynthesis that occurs in that leaf. Different types of plants need different amounts of light. Shade-tolerant plants need less light than sun plants. The main wavelengths of light that are important to photosynthesis are red, orange, blue, and purple. These wavelengths of light are called the photosynthetically active radiation. I apologize. They are called photosynthetically active radiation. Most of the energy that's hitting the leaf from the sunlight is lost. Only 5% of the energy that strikes the leaf is saved. So the electron transport chain is only able to capture 5% of that energy and convert that energy into carbohydrates. The leaf has different ways to increase the amount of energy that can be kept by the plant. In the mesophyll region of the leaf, plants have light channeling and light scattering. These are two main ways that leaves make sure to maximize the amount of energy that they take in. Light channeling is the propagation of light through vacuoles and spaces. It's how light is bounced and propagated through the tissues of the leaf. Light scattering is when the light is reflected or refracted across the leaf to make the path of the light through the leaf even longer. In this, the light bounces around the leaf so that more energy is captured. Plants take advantage of things called sunflecks. Sunflecks are small patches of light that pass between the leaves and onto the ground. So if you have leaves in the canopy, and the spaces between the leaves are sunflecks, and the light from those hits the ground. Understory plants are very dependent on sunflecks. These plants are able to rapidly change the speed of photosynthesis to capitalize on whatever bits of light they are able to receive. Plants are also able to orient their leaves in separate directions if they need to, in something called sun tracking or solar tracking. If the plant orients their leaves parallel to the sun, it is called Hera Heliotropism. This is a Greek word. It comes from the roots para, which means alongside, helio, which means sun, and tropism, which means turning. So the word para-heliotropism is basically they're turning their leaves alongside the sun. So in this, the plants turn their leaves alongside the sun so they have less photosynthesis and less light exposure. Plants do this in some environments where there is too much sun for the plants to take in at once. Plants are also able to orient their leaves in another direction. They orient their leaves in a way called diaheliotropism. Diaheliotropism is also from the Greek. The word dia means through, and then helio means sun, and trophism is movement again. So these are turning, So these plants are turning their leaves so they get sunlight going through them. So these are the plants that want to maximize the amount of light. This is what plants do to maximize the amount of light that they take in during the day. The last important concept here is the light compensation point. This is the minimum amount of light needed so that the amount of carbon dioxide that the plant takes in from cellular respiration is equal to the amount of carbon dioxide that is brought into the plant from photosynthesis. So the amount of carbon dioxide going in is equal to the amount of carbon dioxide going out. In this phase, there is no growth. The plant is merely doing the bare minimum it needs to survive. If the plant wants to grow and recover from illness or a bite or something, it needs to be able to pass that light compensation point. So that light compensation point, where the amount of CO2 in is equal to the amount of CO2 out, is very important. The plant wants to take more CO2 in than it's allowing out. So there's more stuff in photosynthesis than just the textbook view in your biology textbooks. New discoveries in photosynthesis are happening every single day. Just this month, a research team from Washington State University, led by Dr. Helmont Kirchhoff, created a computer model that explained how plants store energy in the thylakoid membrane. Now, the specific energy storage that they're talking about in the paper is the proton gradient during the light reactions. Dr. Kirchhoff's team looked at how ion channels in the membrane can influence the proton gradient and cause changes in the amount of energy available for the plant. So, how we were talking about the light reactions earlier with the electron transport chain and ATP synthase and the proton gradient, they were looking at that, and they were doing exact computer modeling on the specific ion channel proteins that are needed, and they worked that out better. To look at this, they shine light of different intensities on the leaves, and the light excited the pigments, and it started up the light reactions. So looking at the light reactions, it hit photosystem 2, and then photosystem 2 passed the electrons to cytochrome, and then cytochrome passed the electrons on to photosystem 1, and then it went all the way over to NADPH reductase, and it made NADPH. So the more active the electron transport chain and the more active the proton gradient was, they were able to tell how active the light reactions were. So they measured the absorption and the fluorescence given off by the leaf to be able to tell how active the light reactions were. In 2018, researchers from the Imperial College in London discovered a new type of photosynthesis. This research was led by Professor Bill Rutherford and Dr. Dennis Nuremberg. Instead of using red light like the majority of plants do, this one type of cyanobacteria, also called an algae in the paper, used light in the near-infrared spectrum. So it didn't just use the Roy G. Biv visible light spectrum, it used near-infrared. In near-infrared, this cyanobacteria shuts down chlorophyll A, and then it switches to a pigment called chlorophyll F. Now this is important because, looking back, chlorophyll A is the main pigment in the reaction center chlorophyll. So without chlorophyll A, we don't have that main reaction center chlorophyll anymore. So it then continues to do photosynthesis, the plant does, well, the algae, but the absorption is in near-infrared instead of red. Because cyanobacteria lives in shaded conditions, the typical light damage that you would expect to see from doing near-infrared photosynthesis does not occur. These cyanobacteria also have specific mechanisms that are present to protect themselves from damage to this light. Instead of the classic reaction center chlorophyll being responsible for photosynthesis, chlorophyll F, which is typically just relegated to being another accessory pigment in the antenna complex, so it's one of those pigments that are in that circle surrounding the reaction center chlorophyll. Chlorophyll F is actually responsible for photosynthesis occurring. This is a textbook changing discovery. If it holds true for other organisms, it could mean that there is a greater need to consider the accessory pigments when determining the amount of photosynthesis occurring, and also to make more distinctions into what molecules are responsible for which portions of photosynthesis. And lastly, a 2021 study from the Nagoya Institute of Technology in Japan showed that artificial photosynthesis may be possible. This study on artificial photosynthesis was led by Professor Masashi Kato. These researchers have been attempting to find something called photocatalysts. Photocatalysts are some artificial process that can replicate photosynthesis like how plants do. Typically, photocatalysts are some sort of element combination. These researchers use strontium titanate and low concentration niobium to determine the optimal structure for photocatalysts to do artificial photosynthesis. They took strontium titanate and they used different levels of niobium doping, or they used different concentrations of niobium in the solution to determine how much photosynthesis was occurring, or energy conversion, because it's artificial photosynthesis. They determined that moderately doped strontium titanate performed better than pure strontium titanate, and it led to a higher energy conversion rate. With more research into the system of artificial photosynthesis, researchers could develop a sustainable source of energy that could potentially help to power all the electronic devices and all of the devices that we use. They see very much promise in developing this system, and it could be something revolutionary and new in the next couple of years. Thank you for watching this video on photosynthesis. Again, my name is Janae Heiss. I hope everyone learned something, and I'll see you in the next video.